No, we can start from.
First of all, let me say that it's a pleasure to be here and an embarrassment to be showing up late. This actually recalls to me the fact I'm organizing a meeting in Europe in March. It's of course virtual. And we had a conversation many months ago about whether we did do recorded or live talks. And one of the things that came up was, well, what if people screw up the time conversion for the talk? And my response was, what kind of idiot would make a mistake like that? And so, it turns out there are many idiots in the world. I apologize. And I thank all of you for your patience. And I regret, missing all of these fantastic talks due to my own stupidity. Having said that, I wanted to just spend 2 minutes giving you a very brief view into our group and how we operate, and in particular because originally I was going to give a talk about phylogeny, but I realized that several people in this conference had already seen part of that talk, and so I want to present something new and exciting, at least for us, and in particular something that I'd love to get the input of this community on that's related to bacterial evolution. So my lab really has for years focused on bacterial growth. And perhaps the summary of what our lab does is to apply this multi-scale approach where we can integrate both molecular and cellular scale observations into physics-based theories to try to understand the process by which cells grow. And this is, of course, a very big challenge that's pursued by many people. And I think one of the exciting aspects about all of this, though, is it really leads into new technologies because of this grand scope. And so I like to say, our goal is to map growth in space and time, all the way from atoms to cells. But in recent years, we've started to take this conceptual approach where we use both bottom-up and top-down approaches to dissect a complex system and apply it to a very different sort of system. And this is all by way of a very close collaboration with a good friend of mine, Justin Sonnenberg, who's a glycobiologist and studies the effect of diet on the microbiota. The microbiota represents communities of cells that are host associated and that strongly impact health and disease. And what we realized was that really, in order to come to a mechanistic understanding for how these communities function, we needed to apply a multi-scale approach, where perhaps now the units of interest here were cells, and we wanted to bridge to tissues and organisms. And much like trying to understand bacterial cell growth, it really required new approaches from computational modeling, theory, and quantitative analyses. And I think what's really exciting for me is that this Marcel Proust quote of the real voyage of discovery consists not in seeking new landscapes, but in having new eyes, really has a huge amount of applicability to the microbiota, because every new way of poking at this system seems to bring out totally new biology. Of course, the first instance of that was Van Leeuwenhoek, who built the first compound microscope. On the right are his drawings of, in fact, his oral microbiota. You can see an amazing array of shapes and sizes, and in particular here, motility behaviors. And so that too was our first foray into the microbiota, where we realized that everyone was asking questions about who was there based on sequencing, perhaps what was going on based on omics approaches, but the where was kind of this big unknown. And one of the first pictures we took was this one here, where you can see the mouse epithelium on the lower right, surrounded by a thick mucus layer in green, and then all of these bacterial cells out in the lumen on the left, the yellow, the maroon, the red cells. These are all different types that have been stained with probes. And we were able to try and understand simple questions like how diet might affect the organization of all these microbes. And it was fascinating to have this new window, which was now we could start to tell apart individual species. But of course, there's a second aspect, which is also important, that's completely blind to us, and that's the individuality of each one of these cells. Of course, this is a hugely complex and varied environment, and yet we're basically lumping them all into one group. And so today's talk is really about efforts primarily driven by a grad student in my lab, Kim Vasquez, to try to start to tease apart intra-strain behavior within the gut microbiota, and in particular, look at the evolutionary dynamics of individual species. And so this is really a talk that ends up being about evolution and colonization dynamics within a mammalian host. And so the motivation is that every time an organism like E. coli goes into a new environment, there are a huge amount of factors it has to adapt to. The effort in the microbiota side has focused on describing the composition of communities, but it may be equally important how each individual species starts to adapt to the environment, and in particular, the diversity of behaviors that it engages in at the intraspecies level. This is actually something that has been brought home by in vitro evolution experiments, in particular, a long-term evolution experiment by Rich Lenski, where he evolved E. coli for over 60,000 generations. Just to put it in mind, 60,000 generations is basically the entire time course of Homo sapiens. And so this is a really interesting experiment because it starts to bring out factors that are both complex, but also well controlled. What I mean by that is that the environment was kept as constant as possible in this experiment. And yet what you can see here in the plot is that there are certain strains of E. coli within this community now that have developed that coexist with other strains. So the top curve and the bottom curve are coexisting substrains of E. coli, and they persist for tens of thousands of generations before one of them eventually fixes. So I think this is really fascinating because it tells you that life will find a way to develop diversity. And we want to take this to the next level to try to understand it inside a mammalian host, which is the natural environment, or one of the natural environments of E. coli. This is an approach that has or this is a problem that has been tackled by a couple other great studies. In particular, Isabel Gordo and Karina Zaviazab, they applied a barcoding approach where they put in two fluorescently labeled E. coli, a yellow and a cyan. And within a complex community, they could see that E. coli would entrench, and the frequency of yellow versus cyan could go up in some cases or could go down. The selection that was taking place was observed to be related to galactitol metabolism, and they could see clonal interference. And so it was clear that there was a complex mutational landscape for E. coli. Of course, with two barcodes, it's difficult to take a step much farther. And also, this was in the context of many other bacteria. A separate study that was done by Nidis Monak's lab in the context of Salmonella, they put in 8 strains that all had a unique DNA barcode, and then observed that within individual mice, you would see different barcodes arising. And you could also see that, for instance, in the colon, you could see that here the pink and the orange, and here the orange and the blue would persist after a period of a few weeks. And there was also cases where one barcode would come to dominate. But again, this was in the context of of a complex community. And we were really interested in trying to disentangle the complexity of the community from the complexity of the environment, and also avoid problems that might be associated with large bottlenecks by using a much richer set of barcodes. And so what Kim did was to employ a system developed in Steve Quake's lab of plasmid-based DNA barcodes, where we isolated 192 unique barcodes. We split them into 2 sets, S1 and S2. That'll be very important for the rest of the talk. We checked to make sure there were no changes to growth or antibiotic resistance due to the introduction of this plasmid. And then we wanted to ask, okay, can we use these as a tool in the context of germ-free mice? So these are mice that have no resident microbiota until we introduce them, and ask, for instance, what adaptations are acquired during initial colonization and after growth in the host? What's the potential for crosstalk between different hosts? And how much transfer of microbiota does occur both between hosts and between cages? And the barcodes give you a really unique capability to answer all of these questions. I should say this has been a really fantastic coming together of many wonderful scientists, all of them listed here. I'm not going to say all their names, but it's been a really fun collaboration trying to employ all of these new techniques, in particular, new techniques in metagenomic sequencing. So here's the setup. We took mice and split them into 2 groups. One of them acquired set 1 barcodes, and one of them was gavaged with set 2 barcodes. Okay, and we tracked them for 37 days. There was another set of mice that were so-called cross-housed. So we let them equilibrate with either set 1 or set 2 barcodes. And then after 3 weeks, we put them together and allowed them to cohabit for the rest of the experiment. So first, I'll just say that very quickly, the amount of E. coli that we gavaged expanded within 1 day to basically have an expected yield of about 10 billion cells per milliliter. That's typical for commensal bacteria. And so it seems that colonization is very stable and very rapid. We also saw that the barcodes themselves started off at a good state in the sense that they were distributed uniformly across all of the set 1 mice at about 1% per barcode. There were small fluctuations, but basically this was a great starting point for starting to track evolution afterwards. It implies, for instance, that there's no large bottleneck that would immediately select against a large number of barcodes. But things happen very quickly after that. So what I'm showing you at the top are tracking of each of the individual barcodes in some set 1 mice. You can see here that each color represents a different barcode, and we've labeled some of the dominant barcodes here. That's on the top. On the bottom is actually metagenomic sequencing. So we took every sample, subjected it to metagenomic sequencing, and we're able to identify key mutations that seem to track along with barcodes. So already this was a really useful tool for trying to associate the evolutionary trajectories that we observed with individual barcodes. And for instance, you can see here that this FLHE and LAC-I mutation seem to be very closely correlated with this S136 barcode. And I'll keep coming back to S136 for obvious reasons over the next few slides. In particular, I just want to note that anytime you have mutations that appear in 70% of the cells, it's very likely that it appears in the dominant barcode because of the fact that it would be less likely, at least, to be generated independently in different barcodes, although not impossible. What you can see here is that mouse 10, which was in the same cage as mouse 1, has slightly different trajectories, but ultimately ends up at the same state where S136 dominates. In particular, S136 takes over after an initial bloom of S186. And so this is at least a suggestion that the individual mice are not really independent, at least when they're in the same cage. And so I'll also come back to these 2 mutations, the LAC-I and the FLHE mutation, because I'll show you their fitness benefits shortly. However, I want to come back to this issue of initial colonization because I think there's sort of a subtle addendum to this that we're still trying to puzzle through. So I told you the set 1 mice were evenly colonized. The set 2 mice, 2 of them, 12 and 14, were evenly colonized, while mouse 13 and 15 were completely different. In fact, there appeared to be a few barcodes that dominated these 2 mice. And in fact, these 2 mice lost about half of their barcodes in the first day. And so something is different about these mice. We don't know what they are, but we found this really intriguing because it sort of implies that there can be host-specific colonization trajectories. And one of the things that is particularly interesting about having the metagenomic data is that what you can see is that one of the barcodes that was abundant on day 1, due to what we presume is some strong bottleneck, we could associate with the trajectory of a mutation in DNA gyrase. DNA gyrase is very important for dealing with DNA supercoiling, and in fact, of the barcodes that were dominant on day 1, several of them had gyrase mutations. Here's another one that was dominant that had a separate gyrase mutation. We know that these gyrase mutations are not in the initial stock, so we know that they were selected for, but specifically in the context of these mice, in these mice that had the bottleneck. And what we found really interesting is that these gyrase mutations have already been associated with a lot of different phenotypic changes, including broad changes to the transcriptome and conference of multi-drug resistance. And so I just want to pause to draw home the point that what this is saying is that colonization itself can select for antibiotic resistance in the complete absence of antibiotics. And I'll, in fact, show you experiments at the end that draw home how significant that can be through antibiotic treatment. I also want to, so leaving that aside for a second, because we're still find it quite mysterious what's exactly going on with that initial colonization, in part because it's in our minds, unrepeatable without knowing what actually happened to those two mice. But what you can see is that in the set 2 mice, there's actually also a lot more heterogeneity in the final state of the mice after 3 weeks. There's still a set of dominant barcodes. And in particular, if you compare mouse 12 and 13, you can see that the final state is actually really in close correspondence between the two. And in fact, if you look at all 4 mice, regardless of whether or not they had a bottleneck, they end up in practically the same state. And so there seems to be an equilibration within cage that's very important. And in fact, the same group of 10 barcodes make up more than 95% of the population by day 19. So this really inspires the cross-housing setup that I mentioned before, where you combine set 1 and set 2 mice. And what we saw here was that the set 1 mice clearly had a huge advantage. The barcode S136 took over in the set 2 mouse, and while there was some penetrance of set 2 barcodes into the set 1 mouse, the S136 barcode still remained dominant, so indicating it had a higher fitness. And so you might ask, well, what even provides the possibility for the set 2 barcodes to penetrate in transiently then? And the obvious answer is coprophagy. So coprophagy is the fact that mice and many other mammals eat their own poop. And so this inspired a very simple model. It's essentially an island migration model where you assume that barcodes have a particular fitness. You assume there's some replacement of the microbiota each day. And then you ask, how does the environmental exchange dictate the abundance on the following day? And this was work done by a postdoc in the lab, Lisa Willis. It's a relatively simple population genetics model. And so, the next day's relative abundance is a function of the enhanced growth or replacement of each, and then multiplied by the fraction that remains in the mouse, along with the proportion of that barcode that you find in the environmental reservoirs times the fraction of the microbiota that's replaced each day. And this model implies a sort of exponential decay to equilibrium that's not particularly sensitive to exactly how the feces is being consumed. For instance, this R parameter tells you how much does one mouse like to eat its own poop. And basically, this predicts a half-life of, on the scale of days to tens of days, depending on the migration rate. So if we take our data, the data I showed you from the mice a few slides ago shown here on the left, and we simply fit the model to this data, it predicts very well the dynamics both of the incursion of S136 into foreign mice and also the transience of the other barcodes into mice that are already dominated by S136. And it does so predicting that the fitness advantage per day of the dominant barcode is about 75%, but more importantly, that the replacement rate is somewhere between 10 and 20%. So just to let that settle in, what that means is that every day, 10 to 20% of the microbiota does not come from growth in the host. It comes from getting it from outside. Now, of course, if you're thinking to yourself, well, what does this mean for me? I hope that you eat less poop than these mice do, but I think it's a really interesting test case for establishing how important this can be in an organism that clearly has this lifestyle. I'll just show you that the equilibration that we saw in the set 2 mice is very well predicted by the model, in particular because this equilibration timescale for that migration rate is very fast. It's on the order of 4 or 5 days. So I also, in the last few minutes, I just wanted to point out a few aspects of this experiment that I think are very powerful that help to interpret the data. First, we ended up sequencing the genomes of 768 isolates. The main point here is that the vast majority of dominant mutations that we see are in metabolism in green or in motility, shown here in purple. And there are some mutations that are fixed, and then of course there's a diversity across the population. The mutational frequency increases over time, perhaps not surprisingly, so there is continued evolution taking place. So we wanted to ask, well, what drives all this metabolic selection? So we used NMR to characterize the composition of both what's inside a germ-free mouse in its cecum and in its fecal pellet, to ask, well, what nutrients would be available for an incoming bacterium? And you can see here that raffinose and stachyose are 2 trisaccharides that are very abundant. It turns out that Stachyos is not, neither one of these is consumed by E. coli, by wild-type E. coli. But what you can see here is that the LAC-I mutation that I mentioned before actually enables the evolved clone to consume raffinose, whereas the wild type doesn't show any growth at all. And so this suggests a very simple picture, which is that available nutrients are a strong and perhaps the strongest selective force in vivo. And so just to illustrate how clean potentially that result is, one of the things we wanted to do was to replay and extend evolution to sort of address this fact that evolution is continuing, and also that there perhaps is a very strong and simple driver for evolution. And so one of the things that we found in a new experiment with completely new germ-free mice is that the same mutations, the LAC-I and the FLHE mutation, appeared in the second round of experiments. And so, in fact, despite the fact that this is a very complex host, the evolutionary clock is replayable, at least in this context, or at least in these mice. We also were able to take the S136 strain, remove its barcode, and rebarcode it with a new set. And you can see here that clearly there's a new set of dynamics that is taking place. And so again, this is further evidence that there's a huge evolutionary potential for these strains beyond this large fitness advantage that it acquired in S136. But I think one of the things that's really exciting is that I told you that in the first experiment, we get these two dominant mutations that appear. In the second experiment, we also get these two dominant mutations that appear. And so one of the things you can do is to ask, what happens if you were to compete the two? And so we actually took these two sets of mice, combined them together, and what you find is, in fact, that with the same microbiota replacement rate of about 20%, that the dynamics of the first set of barcodes, which are shown here in orange, and the second set of barcodes shown here in blue and purple, actually now end up equilibrating. No one takes over, and that it reflects the fact that they have the same mutations, essentially. They have roughly the same genomic content, and so they have equal fitness in the gut. And so I think this is it's exciting because it really suggests that there is a prescribed trajectory, at least in the context of this organism coming into a totally new landscape. Whether that continues in the future, it's probably unlikely, but it's exciting to think that at least on the timescale of months, that there's a predictability to evolution. So with that, I'll just conclude by saying that I think the barcodes really give you a unique power, especially coupled with metagenomics, to reveal the sources of rapid selection and fitness gains, but also to integrate that with information about how external reservoirs and invasion affect the population. We think it's really exciting that there is the potential for selection during initial colonization that can have really pleiotropic effects like antibiotic resistance. I didn't show it here, but we were also able to use the metagenomic data to address questions about genome replication frequency and show that really the growth rate of these organisms inside the gut is very low once they settle down. But we think there's a lot of potential for barcoding and metagenomic sequencing for quantifying gut ecology. So excited to talk with any of you about this and other future aspects. So I'll just this is a joint group meeting, a joint lab retreat that we had a couple years ago with Brett Chang and Will Ludington's lab. I have a really fun group of people in the lab that it's really a joy to work with, and it's sad that COVID prevents us from seeing each other every day. I think I've thanked all the collaborators involved, so I'll just stop there and take any questions.
Okay, so thank you very much, Casey, for this great talk. We already have some questions in the chat. So we have one from Eric van Nimwegen. He's asking, what are the holes in the bacterial soup? I think this refers to your, the picture you showed rather at the beginning of your talk where you had all these dense bacteria.
Yeah. Yeah.
Right. Yeah.
Yeah. So, okay. I have two hypotheses. There's the boring one and the exciting, well, the semi-exciting one. So the boring one is that it is a function of preparation. And perhaps you've ripped out part of the sample as it was prepared for imaging. That is the kind of go-to answer by reviewers. But we actually think that's unlikely. It may be true. That doesn't really bother me very much because of their scale. But we don't actually think that's true, in part because if we do deep 3D imaging, it looks like it's very self-contained. I mean, it looks like we didn't like tear anything out of the sample or anything like that. So what else could it be? Because it literally is like the dark matter of the lumen. It very easily could be gas bubbles. In fact, I'll just regale you with a story where we've developed an in situ imaging approach for looking inside the gut of intact Drosophila flies. And I found this very exciting because we could then do time-lapse imaging of microbiota dynamics. And the very first experiment we did, we put the fly on the microscope and everything was in focus and we were about to start recording, and suddenly everything went crazy. It looked like there was some sort of massive bubble that passed through the whole sample. And then suddenly nothing was in focus. We didn't know what we were looking at. And we sort of stared at each other. And my student said, I think we have just observed the first in situ fart. And of course, there's a lot of gas production by these organisms. So that would be my best guess, but I'd love to entertain any other ideas.
Thanks.
Okay, cool. So we have another question in the chat from Michael Manhart. He's asking, especially since you see the same barcodes invading in different mice, do you know if some of the adaptive mutations were already present at low frequencies in the initial barcoded population?
Yeah. So I think, first of all, some of these mutations are quite complex. I didn't say this in detail, but the FLHE mutation is actually a 16 kilobase deletion of almost the entire motility operon. And the LAC-I mutation is actually a 4 base pair deletion. And so I'm bringing this up because the fact that they are not point mutations at least makes me think that they're less likely to be present in the initial inoculum. They are not detectable in the initial inoculum. That doesn't mean they're not there, of course. And so what I can tell you is that what was very comforting to us, I also didn't mention this, but in the second experiment, we were specifically worried about this aspect. And so when we recolonized the mice, we left out the strain that dominated in the first experiment. And so by necessity, a new strain ended up dominating, but it had exactly the same mutations as in the first experiment. So I think it's unlikely that they were there at the present, because if they were there, then that strain would have been able to compete in the first experiment as well.
Can you just quickly say that LAC-I mutation, did you only see that once or did that show up in multiple mice?
That showed up in multiple separate experiments.
Wait, how can that be? I mean, it breaks LAC-I, right? It's basically it knocks out the repressor, right?
So probably not. It actually is a mutation that, okay. So this is where my head started to hurt, because what I thought I understood about the numbers that dictate evolution was well, okay. Let me just tell you what we observed. I told you that this enables cells to grow on raffinose. And so one of the things we tried to do was to evolve a raffinose mutant in vitro. Now, we were completely unable to do this through a ton of different efforts that I can tell you about if you want. But you don't need to believe me. We then found a paper where someone tried to do this and also failed, but then successfully evolved raffinose mutants through a much more convoluted approach where they had to upregulate alpha-galactosidase and all sorts of things in order to do that. So you might say, okay, well, is this mutation really hard to acquire? Well, no, it's not, because it was observed in a paper from Tim Cooper's lab where they did in vitro evolution, where E. coli was switching between glucose and lactose every day. And so there are environments where this mutation can pop up quite readily. It clearly pops up in mice, and it clearly pops up in this oscillating carbon condition, but we can't get it to happen in the context of what we think is the dominant selective force in the gut. And so I don't know what to make of that. But we do see it. We see it appear in two completely different set of mice that were separated by months, if not years, in experiment.
Okay, cool. I mean, we will have to wrap up rather soon, but I see that several people have their webcams on. So, if you wish to ask a question, Please go ahead.
Yeah, I have a short question. Thanks for the very nice talk. So I was wondering, with this barcoding assay, you mentioned that you follow, I think, about 200 different strains.
Yeah.
In vitro assays can really follow hundreds of thousands of strains. So where is the difference? Why is it much harder in mice?
Yeah, it's definitely not hard. Well, okay, I should say it could be harder in mice. You're definitely on the right track, though. So the reason we did this was because it was a very well-characterized set of barcodes to start with. And so we thought it was a good trade-off as a first experiment. For instance, there's always a concern about, as other questions brought up, pre-existing mutations that might affect phenotypes. And so this enabled us to really, for instance, check check growth, check resistance. I didn't have time to get into it, but we also treated these mice with ciprofloxacin. That ended up being totally accidental, that it overlapped with the gyrase mutations. And the cipro came, months after the gyrase mutations arise. So there was no possibility of cross-contamination. But I think you're exactly on the right track. So we've actually done these experiments now with Bacteroides theta iota omicron, which is one of the model organisms for the human gut. And there we used tens of thousands of barcodes. Now, there's an interesting challenge technically that I'm happy to talk about more, which is associated with how do you have enough DNA inside a fecal pellet in order to avoid problems based on PCR amplification jackpotting, basically, in order to be able to have enough DNA to sequence later on. And so that's an interesting question to deal with. Places fundamental limitations on how many barcodes you can use. But we think we've got pretty good data associated with this. And then, of course, the mice introduce an additional wrinkle, which is, with more barcodes, you get to see more deeply, but also then there's more potential for just random fluctuations that can eliminate barcodes, and that has to be taken into account.
Cool, thanks.
Okay, cool. Do we have any other pressing question? No? I mean, I see Daniel has a comment rather than a question, but okay, it can be moved on to our social in the gather room. Before we leave for the social, I think Michael has an announcement.